We're here at the University of Miami Experimental Hatchery, where we're in the midst of our first leg of an experiment to determine the effects of tagging on juvenile mahi-mahi. This is going to be extremely important for an upcoming cruise that we have this summer, where we're going to be tagging large mahi-mahi with pop-up satellite archival tags from wildlife computers. These tags will give us very important information on how the mahi-mahi are utilizing their habitat, as well as where they travel after leaving the Gulf of Mexico. We're tagging small fish with an identical tag scaled to a smaller size that simply doesn't collect any of those movements. And with that, we're trying to figure out what the cost associated with actually carrying the tag is for the fish. For the first part of this experiment, we're going to be using swim tunnel respirometers to measure the metabolic effects of tagging on the fish. To do this, we're going to take fish of approximately 25 to 35 centimeters, and we're going to be running a paired experiment with one tagged fish and one untagged fish that have both undergone the same handling stress. After the fish are fed, first thing in the morning, they're going to be removed, and one is going to be tagged and the other is going to be subjected to the same handling stress. They're then going to be placed in recovery tanks for approximately 8 hours. After the recovery period, both fish are going to be moved to our swim tunnel respirometers, and general length information is going to be collected on the two fish. The fish are going to be allowed to acclimate overnight, and then first thing in the morning, after we've determined that the fish have been unexposed to any kind of outside stimuli, our trial is going to commence. At this point, we're going to increase the speed at which the fish are swimming in our swim chambers in 20-minute intervals. This will allow us to determine the maximum sustained swimming speed of a tagged fish versus an untagged fish, as well as the oxygen consumption of that tagged versus untagged fish. This study will allow us to compare the swimming abilities of tagged and untagged fish. The importance of this is that it will allow us to better interpret our data that we collect from our larger fish this summer.